Okay, so please watch this video on DNA replication and then come back to me. So DNA replication is the name of the process that we've already talked about that occurs during the S phase of interphase. It's copying DNA. That's all that DNA replication is. And what we're making are identical pieces of DNA, because you need identical pieces of DNA in order to do cell division, whether or not it's mitosis or meiosis. So the who, what, when, where, and why of these is it's going to occur in interphase. So it's going to occur in that period of interphase, the S phase, after the cell is grown. Then it's time to divide the DNA to prepare to make a new cell. So it occurs during interphase. And where it's going to occur in the nucleus of eukaryotic cells like yours and mine. But if you're talking about, let's say, some bacteria that doesn't do mitosis, instead it does binary fission, it still has to copy its DNA before it splits, and it would occur in the cytoplasm because it doesn't have a nucleus. So we're going to look at how easy DNA replication is on paper. Very super easy. All you have to do is remember your base pairs. That's it. Okay, DNA and DNA replication is considered semi-conservative. And what that means is that every time you go to make a new cell and you replicate your DNA first, then the DNA is going to split, and the original strand or the old strand, the parent strand, is going to serve as a template to build the new strand, which is called the complementary strand. So that means that each new piece of DNA that you make, each new double helix that you make, is old piece, new piece, old piece, new piece. So you see a little bit of that here in three different views of this, but you could see on the left there, the parent versus the new strand. They call it like the daughter strand. I don't know many people use that. It's kind of cute though, isn't it? But most people call it complementary. So if you see that on that second piece there, and then if you look at the third one, you could see 3 and 5. And what does that mean? 3 prime and 5 prime. So what it's talking about is top versus bottom. So 5 with that little apostrophe beside of it's called 5 prime. That's the top. The 3 prime is the bottom. So what that means is if you look at that third picture, and you can't really tell it unless you look at the 5 and the 3, if 5 is top and 3 is bottom, that means that one strand of DNA is right side up and the other side is upside down. So you could see that DNA replication doesn't really start at the very tippy top. It starts in locations and places called primers, and then that's a place where this replication starts. But it does start, as you could see in each of these, by a separation of the two strands of this double helix, which is called unzipping. And then once that happens, you can see that middle picture here that the complementary strands, the matching pairs, come together. And what does that is a type of a couple of enzymes involved here. So one enzyme is involved in the separation. That's a helicase. It's given this DNA heck and it's taken it apart. And then the one that does the joining back together and some proofreading is called DNA polymerase. So it's forming a polymer, it's forming that DNA molecule. So one more little practice here before I set you on your way with a little piece of paper in front of you and some practice and some colored pencils and all that jazz. So what we have here is one strand of DNA, and we have the bottom of it. So the 3 prime is the bottom and the 5 prime is the top. And replication occurs from top to bottom. So it goes from 5 to 3. And what those 5 and 3s come from, that comes from the sugar. Oh, it didn't mean to do that. It comes from the sugar. I've got to be very careful. My mouse here is very sensitive. But The 5' is the fifth sugars up at the top. The 3' is the third sugar. So each point in that deoxyribose sugar, if it doesn't say a different element, then it's going to be carbon. You just assume it's carbon. So the fifth carbon's at the top or the third carbon's at the top. All right. So I just wanted to give you a chance to practice these with me before you do them on your own and turn them into Miss Gillum. So GAC, G goes with C, A goes with T, G goes with C. Boom. The next three, and again, I'm doing these in threes. It'll make sense later why I'm doing it in threes. TTC, T goes with A, T goes with A, and G goes with C. So all turkeys can gobble. Next three, CGA. C goes with G, G goes with C, A goes with T. And then the last three, TAC. T goes with A goes with T, and C goes with G. I'm clicking, and there you go. Okay, so I'm going to give you some things to do to practice this, but the first one, and you've got a couple of things, but I'm just calling this the first one, is DNA replication illustrations. I call it the DNA color sheet or the DNA color worksheet because you're going to be using some colors. Okay, I'm just going to read this with you. It says, below are the steps of DNA replication, which take place during the S stage of interphase. In each box, read what is taking place and follow the given directions. Pay attention to the specific coloring directions. Then it says, use chapter 12, section 2 in your book to help you. I forgot to remove that. It has nothing to do with us. Okay, so in the first one there, this looks like a little storyboard of DNA replication. So the first part of this is a double helix molecule. Label the following: nitrogen base pair. So there's not a lot of space up there. Maybe you could just put NB for nitrogen base. Sugar phosphate backbone. Not a lot of room for that. Maybe you could just put S-P or S/P. And then the color, it says draw a thick blue line over each helix strand to represent the original strand. So this is your original DNA before you've done anything to it. All right, step 2. Unwound the DNA molecule. So remember, this thing's twisted because of those hydrogen bonds, so it has to unwind. Now you're to label the following: the deoxyribose sugar, so maybe you could just put S. And remember, your nucleotide looks like a shaka, P pinky phosphate, connected to sugar, and then the business end, the thumbs up, is the bases, which are the code. For P phosphate, and then the missing bases, starting up there with that G, all you have to do is put in who G pairs with. Does it pair with A? Does it pair with T? Does it pair with C? Hopefully you know, or you can go back and look at your memory devices. Apple Tree Car Garage, all turkeys can gobble. For the sugars, we're going to color them orange. A's are green, T's are pink, C's are red, G's are purple. If you're colorblind, make sure you read the little list there. Or if you just don't like any of those colors, just change your key down there to what color you want to use. Okay, the third step, this is an unzipped DNA molecule. So the missing bases, we're going to fill those in just like we did on the last one, and then we're going to put in the DNA helicase. So this is the one that I said does the breaking apart. So there's a little bubble there for you to put in this helicase and then do your coloring. Third step, the actual replication. So we're going to label the newly added bases. So again, just put the missing bases in, and then the DNA polymerase. So this is the one that joins the nitrogen bases together, and this is the one that proofreads and makes sure that they go in the right spots. So you've got 2 spots there for DNA polymerase, so maybe you could just put DP. And then color coding, color the original strands blue and the new strands yellow. And you'll do the same thing with the last one, which shows you your 2 new identical pieces of DNA that are semi-conservative, old piece, new piece, parent and complementary. Just color the original blue and the new yellow. So this is the first part of the work that I'm going to have you do with DNA. Good luck. Take your time.